Okay, thanks for the invitation. I hope you are ready for a bit more. But now on the DNA side of things. So I try to be concise. So I'm Michał Grabowski from University of Łódź, and I also represent the Polish Barcode of Life and the International Barcode of Life and a couple of these European projects that I will talk about a bit more. So first of all, this will be very, very short, just a short recall on DNA barcoding, just to make sure we are on a common ground. Then a short review on the DNA-derived biodiversity data in Poland, then Polish Barcode of Life, core biodiversity genomics projects that are going on in Europe, and perspectives which are, of course, bright. So why DNA barcoding? Just shortly. We have this taxonomic impediment, we have this biodiversity that we have no idea what it is still, and then the traditional taxonomy works too slow to overcome this problem, and we need something that could help us, and this is the DNA barcoding. So DNA barcoding, you know probably very well this face, which is Paul Hebert from University of Guelph, Canada, and he got this idea that we can use the that you can use the short DNA fragments just to tag different species and then to identify them this way. Okay, so probably it will not be that fast that I imagined. Okay, and so one thing that we need to mention about is basically that we have DNA barcoding, which is providing these genetic tags specimen by specimen, and ideally they are taxonomically identified, and then we are creating the reference, the DNA barcode reference library. And another approach is the DNA metabarcoding when we work with bulk samples, and eDNA when we work with the traces of DNA in the environment. And to get an idea what is really there, we need to have a reference library to identify the short fragments that we have from bulk samples and also from eDNA. So production of DNA barcodes, and especially the reference libraries, is managed by International Barcode of Life. Now I think it approaches 40 countries associated by their national nodes, and the national nodes are trying somehow to coordinate these efforts in each country. And the overall goal of IBOL until 2045 is, of course, very ambitious. Every species is known, every interaction is revealed, every species is monitored. Yeah, it's not really realistic, but why not to try? We have no other choice. So then all this data that are produced, meaning the DNA barcodes, they are stored and analyzed in something which is called BOLD systems, or just simply BOLD. And you can access it either via the original website, the BOLD system.org, or the European mirror that we have since, I think, last year, which is BOLD System EU. And then you can explore what is there by different taxonomic groups, species, areas, and so on and so on. And then also you can have your own profile and you can upload and analyze the data. So if it comes to Poland, I just checked recently that in GBIF we have around 7.5 million of georeferenced records regarding the biodiversity, and that's more or less the distribution. But if it comes to the biodiversity DNA-associated records from Poland, they are, of course, far less. It's around 130-something thousands. So, and how did it change along the time? So, as you can see, until 2010, nothing really was happening that much. And then we have this growth which goes on since 2016. And then we will focus on that a bit more in a minute. So, and what is the taxonomic representation of this biodiversity DNA-derived data? So, as you can see, there are fungi, plants, protists, bacteria. So this is the majority. But in a minute, you will see why. Okay, and the main provider of this data from Poland is International Nucleotide Sequence Database Collaboration, also because it's basically agglomerating the data from other sources also and it pipelines it to GBIF. Oops. Ooh. Okay. Ah, it's probably because the pen drive was. We need to have the pen drive back. Nie, to nie ten mały. Yeah, sorry for that. I uploaded the presentation last night, but then I made a couple of changes. And usually the better is an enemy of the good. Apparently this time as well. Thank you. So we know now where the data comes from and that how this data from EN-SDC accumulated over time, the state for 2000, and then the state later on. It grows on continuously. All right, so already we heard about the MDT. So just a short info about it. And I think it's worth to encourage everybody to put this DNA-based data in GBIF. And you can do it first by training with the MDT sandbox. It's like not a real repository, but you can train how to do it. Then when you are sure that you can do it, you go to GBIF conversion MDT. And all that finally ends up in the GBIF-PL metabarcoding datasets, if the data come from Poland. We don't have that many datasets so far. There are 8. So still a lot of things to do. And the first one shared was from University of Warsaw, and that was this metabarcoding of fungi in post-industrial soils. So, okay. And then the second about the ants and the microbiome. All right, but if we exclude this eDNA data, then we end up, with the pathetic 18,500 of barcodes, but quite well distributed all over Poland. Actually, this red dot here is a cabin in the forest that belongs to me. So it's like a little biodiversity hotspot in Poland, very well barcoded. Okay, and that's how the DNA barcode number grows over the years. So as you can see, it started to grow rapidly after 2020, and there is a reason for that. And we will also talk about it in a minute. And if you look at the taxonomic representation of the data from the DNA barcodes, you can see that they are strikingly different from the general pattern. Why? Because everybody loves insects. Probably that's one of the reasons, but another reason is that you can quickly collect arthropods and insects. And why not plants? I mean, look at, there's just a small share of plants and fungi. Why? Because they are much more complicated to barcode. Because you don't use one marker for them, but more. Also, the laboratory procedure is a bit more complicated. So insects are far easier. Okay. And here we go to Polish Barcode of Life that was to be established in 2019, but then the pandemic came and everything collapsed. So we could start in 2022, and then you can join the points. So this year, and then the accumulation of the DNA barcode data in Poland. It's not a coincidence. It's not a pure coincidence. So the Polish Barcode of Life is a node of International Barcode of Life, which is hosted at the University of lots in my department. What we are trying to do, we are trying to build publicly accessible, comprehensive DNA barcode reference library for the Polish biota, starting, of course, with animals and inverts, because we are specialists in inverts, aquatic fauna, dark taxa, but also myriapods, dipterans, orthopterans, saproxylic insects, some groups that are not very big at the beginning, but you have to start somewhere. And we are associated with Polish Biodiversity Information Network. That's our, let's say, somehow institutional route. And then this way also by GBIF. And we get the data and help, and the network is mostly the scientific institutions, but also the protected areas like national parks and citizen scientists, which are a wonderful group of people to work with. When we started, or before we started, from Poland you had only not even, let's say, 5,000 barcodes, 800 barcode index numbers, which are molecular operational taxonomic units used by BOLD, and just 3,000 sequences with some species assignments. Right now, it's way better. So we have more than this 18,000 sequences, then more than 6,000 beans, and more than 12 or nearly 13,000 of sequences with species assignment. It's still not good, but much better than it was. And the recent idea and recent thing that we are working on together with the Polish GBIF node and also with the Polish Biodiversity Information Network is to link the data from the digital catalog of biodiversity of Poland with the DNA barcodes that are coming from Poland. First, we start with 1,000 species. This is achieved, so I hope it will be published soon. Talking about the projects. So the biggest, as apparently it is, the biggest investment ever in biodiversity research in European Union was the Biodiversity Genomics Europe project, which was funded by the Horizon Europe project, Horizon Europe framework, and it was 22 million euro. So that was held from 2022 to 2026, hosted by Naturalis, and it joined these two networks, IBOL Europe and ERGA, the European Reference Genomic Atlas. Together, there were 34 institutions from 20 partners, 20 partnering countries, with 3 objectives: to build the capacity, production, and then application of both the DNA barcoding and derived techniques and genomics into biodiversity monitoring. It was a very complex project with many work packages and so on. We somehow succeeded, but it was not easy. If it comes to Poland, then this is the geographic distribution of the data that we provided to BGE. So both for reference libraries, but also the meta-barcoding of pollinators, the meta-barcoding of high mountain fauna from the Tatra mountains, and also the dark taxa. So those little small insects that nobody cares about and nobody knows what they are. These are the dark taxa. Not only insects, but generally all those species that are somehow escaping from taxonomic analysis. What we also tried to do, and this is an example from Poland, but what we tried to do in BGE is to engage taxonomists. So that was a taxonometon that we did for the dark taxa, and here in this case it was for all these little flies, and that was amazing to see all these people, the specialists in various families of flies that came together and sit under, or above these samples, and they identified the materials. We also did several trainings on ONT barcoding and data analysis, and we will still do more. And of course, there were quite many papers produced within that project, but I would like to drag your attention to a couple of them, which are somehow providing the perspectives for biodiversity genomics in Europe. So one which is about the engaging of community science, so citizen scientists, and the two that are providing, first of all, the perspective for the development of the DNA of eyeball Europe, and the other one, which is more or less a set of advices of how to build the national initiatives. And this is in Polish, but it says good practices in digitalization of zoological collections. That was a collective effort from many partners of KSIB. And also it includes a chapter for collection managers on how to get the DNA data out of the museum material. It's a free publication, I think, also interesting. And from another European project, which was on eDNA, it's called eDNA Aquaplan, it ends this year. So that tries somehow to overview what is available in repositories regarding the eDNA data from the aquatic environment and, let's say, set up some standards. And the follow-up project of BGE was, or is, BGE Plus, that started just in May, and last week we had a kickoff meeting in Naturalis. It's again the hosting institution. It will be continued until 2030, and a part of eyeball Europe and ERGA, it also includes SETAF, which I think it's a really good development. So again, it focuses on large-scale DNA barcoding, whole genome sequencing, and then also taxonomic expertise and reference material, because without that, you don't have good reference libraries, and without the good reference libraries, not much can be done. And the BGE Plus is seen as a stage of development into European research infrastructure. So the Biodiversity Genomics Europe can become part of one of the existing infrastructures, possibly LifeWatch. So the consortium is much smaller. So it's 22 institutions, 12 million euros, still quite a big investment. And these are the goals, and why it says widening participation, even if the consortium is smaller. It's because there is an implemented cascade funding mechanism, and this is very important, because this project will work as a small grant agency. So far, the call will be announced probably a year from now. We are still thinking about exactly the priorities, but more or less it will be production and application of reference barcodes in genomes. And we are planning to fund at least 42 projects, 60,000 euro each. Of course, if the projects will be cheaper, then probably there will be a higher number. So keep an eye on that, because if you want to get some funding for the DNA-related data production, I think it can be a good source. And one more thing, which is very important, and I'm just coming to the end, is other follow-ups of BGE, which are the cost actions. I think you are aware of the cost actions, so these are not the real scientific projects, but the networking projects. So generally what was identified, and it's not hard to identify, is that there is a lot of fragmentation going on right now in the biodiversity DNA-based research. So it was told here. We have the taxonomic impediment, we have this fair and care fragmented landscape, and then, of course, the differences in lab procedures, and of course, the differences in how the diversity is measured in different strategies, like a water framework directive, marine strategy framework directive, and so on. So what we need is some kind of unification, and we need to get from this fragmented genomic research to something which is more coordinated, more governed. And there are these three cost actions that just a couple of weeks ago we got an information that they were funded. And in each of them, we have the Polish institutions among the proposers, which is very nice. So tomorrow they will be public, these results. So you have the eDNA for policy. That's the action number, which is focusing on integrating environmental DNA into policy frameworks. Then we have Consensa. This is managed mostly by ERGA, and that's consensus standards for applied biodiversity genomics, meaning genome-based. And then we have the DNA BARDIF, which is more or less initiated by IBOL Europe, and it's about strengthening and widening the consortium. So altogether, more than 40 countries. These are the goals of these projects, but we'll not focus on that. But also another important thing, everybody can contribute to this. As soon as it's made public, you can, these are just the examples. But how the scientists can contribute? It's either by joining the management committee that you apply to the ministry that delegates you as a member to the managing committee of the cost action. You can apply for working group membership. So it will be, of course, made public what is the structure of each of the cost action, what it focuses on. And there are different working groups on different subjects. You can basically join it, and there are funds for like conferences, but also workshops and so on. Then this is fantastic. Short-term scientific missions. It funds the research stays of up to 3 months in different institutions within the cost action, especially for the scientists from the ITC countries, so inclusiveness target countries, and also for young scholars. This is really good. And then you have the pilot case studies in different cost actions that you can again see and decide whether you are interested. BioBlitz events. There will be some conferences and so on. So these are 3 cost actions, quite a lot of funds, and quite a lot of good chances to participate and strengthening the biodiversity information. Okay, and just really finishing everything. It's a very new project that we got from our Ministry of Science and Higher Education in the Science for Societal Development projects. It's called BioCrosta, which is about getting a snapshot of biodiversity in different protected areas. And that's the budget. It's not a lot, but also not that small amount. And we so far have in the network 40 protected areas in Poland, 16 national parks and 24 landscape parks. We will do the maless trap meta-barcoding, but also barcoding for reference libraries, soundscaping, and also a lot of engagement for the visitors, the scientific staff of the national park. So basically, not only to get the idea what lives in these national parks in terms of arthropods, but also to educate the society of what is the value of national parks in protecting biodiversity. So thanks for your attention. Thanks for being too long.